All right, let's jump right in. Welcome to today's explainer. Get ready because we're going to completely change how you view the microscopic world inside you. For way too long, textbooks have made the cell look like this. Well, kind of boring, static, microscopic blob. But today we're looking at the cell for what it truly is. A thriving, highly complex, and heavily defended metropolis. Welcome to the cellular city. Now think about any major city in history. Its survival entirely depends on its borders, right? If a cell is this bustling city filled with precious cargo and vital machinery, how does it decide what gets in and what stays out? How does it keep the toxic invaders at bay while welcoming in the vital supplies it needs just to keep the lights on? You guys, this isn't just basic biology. This is a high-stakes border control issue. The literal survival of our cellular city depends entirely on its perimeter defense.
So let's dive into this. The ultimate city wall is the cell membrane, or biomembrane. At first glance, it might just look like a colorful mosaic, but this is an incredibly complex, dynamic system. Looking at the data, it's primarily made of a massive amount of lipids, about 45 to 50%, alongside an equal amount of proteins, and a smaller chunk, roughly 4 to 8% of carbohydrates. To put that scale into perspective, there are about 5 million lipid molecules in just a tiny 1 micrometer square area of this bilayer. I mean, it is absolutely packed.
Moving on to part 1, building the wall. Let's look at the cellular bricks and mortar.
To really appreciate how this highly specialized perimeter functions, we've got to zoom in. The secret to this entire structure comes down to one brilliant chemical property, defined by a fantastic word, amphiphilic. Say it with me, amphiphilic. These are molecules that essentially have a split personality. One end of the molecule is hydrophilic, which means it's polar and water-loving. The other end is hydrophobic, meaning it's nonpolar and, well, absolutely terrified of water. It's water-fearing. This chemical split personality is the exact reason our cells don't just dissolve into mush.
And here are the most abundant bricks in our wall, the phospholipids. See, they have that water-loving phosphate head pointing outwards and 2 water-fearing fatty acid tails pointing downwards. Now, because our cellular city is surrounded by water on the outside and filled with watery fluid on the inside, these phospholipids naturally organize themselves into a double layer, a bilayer. The hydrophobic tails hide from the water by sandwiching themselves in the very middle, while those hydrophilic heads bravely face the watery environments on both the outside and the inside. It's literally a self-assembling barrier. But, you know, a wall made purely of these lipid tails would actually have a major problem. If it gets too cold, the tails could pack together tightly and freeze up, or crystallize, which would cause the membrane to shatter. No way we want that. That's where cholesterol comes in. These tiny, rigid structures are tucked right in between the phospholipids. There can actually be up to 1 cholesterol molecule for every single phospholipid. Cholesterol acts as this crucial stabilizer. It prevents the membrane from crystallizing, keeping the whole wall perfectly stable and intact, no matter what the conditions are.
Okay, so what about the outside of the wall? Well, the cell has this heavy-duty protective armor called glycolipids. These are lipids with sugar molecules attached, and they're completely asymmetrical, meaning you'll only find them on the outer layer of the wall, facing that harsh extracellular environment. They protect the membrane against destructive stuff like low pH and degradative enzymes. Plus, they act kind of like ID badges, allowing for cell recognition and helping cells stick to one another.
Which brings us to section 2, a fluid moving crowd. Let's talk about membrane dynamics.
To really grasp membrane fluidity, imagine our phospholipids aren't static bricks, but rather a dense moving crowd of people at a concert. They are constantly shuffling side to side, exchanging places with their neighbors in what we call lateral movement. And this lateral movement happens all the time. However, what is incredibly rare is a flip-flop, like a lipid from the top layer suddenly diving down to the bottom layer. That almost never happens. So the wall is fluid and flexible side to side, but it maintains a strict top and bottom organization. Moving right along into section 3, meet the protein workers.
Membrane proteins are the true operators of the cellular city. Some act as receptors, detecting signals from the outside world and transmitting that info right into the cell. Others act as anchors, establishing connections to keep the cell in its proper shape. But perhaps the most vital workers are the transporters and channels. They're basically the highly selective guards and cargo managers stationed at the city gates, deciding exactly which molecules are allowed to cross that lipid bilayer.
And that leads perfectly into section 4, the gates in action. Passive versus active transport. So the absolutely crucial point here is understanding the difference between two main systems, passive transport and active transport. If you're taking notes for an exam, definitely star this section. Passive transport is like riding a bike downhill. It requires absolutely zero energy consumption from the cell. Molecules just flow naturally from an area of high concentration to an area of low concentration. Active transport, on the other hand, is like pushing a heavy boulder uphill. It requires the heavy lifting of cellular energy, specifically a molecule called ATP, to force molecules against their natural concentration gradient.
Let's look at passive transport first. Some molecules are like VIPs at an exclusive club. They don't even need to use a gate. Small, non-polar, hydrophobic molecules like oxygen gas and carbon dioxide can just casually stroll right through the phospholipid crowd via simple diffusion. Because they don't have an electrical charge, those hydrophobic lipid tails don't bother them at all. They just naturally diffuse downhill into or out of the cell where they're needed, completely free of charge.
But what about slightly larger molecules, or molecules with a charge, like water or glucose? Yeah, they can't just slip through the lipids. They need specific gates. Here we're talking about channel-mediated and transporter-mediated passive transport. For instance, water uses special continuous pores called aquaporins. Glucose uses specific transporters that undergo conformational changes, which is just a fancy science way of saying they change shape, to let the sugar inside. But listen closely. Even though they're using a specific open gate, they are still flowing from high to low concentration. Therefore, it still costs the cells zero energy. It's entirely passive.
All right, let's pivot and see how this builds into active transport. Sometimes the cell desperately needs a molecule that's already in really low supply outside the cell, or it needs to pump out waste that's already highly concentrated outside. It has to force cargo uphill. To do this, the protein transporter must burn ATP, the cell's ultimate energy currency. The ATP breaks down, releasing a burst of energy that forces the transporter protein to physically push the molecules against the natural flow.
And the absolute most famous example of this is the sodium-potassium pump. This pump is the ultimate heavy-duty guard at the city gates. Here is exactly how it works in 3 steps. First, ATP energy binds to the transporter protein. Second, this energy is used to grab sodium ions from inside the cell and force them out into the extracellular matrix, even though sodium is already highly concentrated out there. Third, the pump reaches out, grabs potassium ions from the outside, and drags them inside the cytoplasm. So out goes sodium, in comes potassium, over and over, all powered by ATP.
Now you might be wondering, why does this matter? Why spend so much precious energy doing this heavy lifting? Because by constantly fighting the natural crowd and keeping sodium out and potassium in, this pump creates a difference in charge between the outer and inner membrane. It creates a literal electrical potential. And guys, this tiny, microscopic electrical charge is exactly what allows your nerve cells to fire signals and your muscle cells to contract. Every thought you have and every single movement you make is possible because of this tiny active transport pump tirelessly working at the city gates. In fact, your cells spend up to 1/3 of their entire energy supply just running these sodium-potassium pumps. Isn't that wild?
So there you have it. The cell membrane isn't just some static bubble. It's a fluid, moving crowd of phospholipids held together by cholesterol, armored by glycolipids, and fiercely guarded by protein transporters, making life-or-death decisions every single microsecond. Without this dynamic wall, the cellular city falls, and life as we know it completely ceases to exist. It really makes you wonder, what other microscopic, perfectly engineered cities are functioning deep inside of you, keeping you alive right now? Stay curious, keep studying your biology, and I'll catch you in the next explainer.